So this is going to be more of a research talk than a course, than a lecture really, but the idea is to tell you about some concept of stochastic dynamics, let's say, that could apply to cellular organelles. And I couldn't attend this morning, but I'm sure you've heard already many things about organelles from Hyman in particular. So in my case, I'm more interested by membrane-bound organelles. So this is a typical sketch of a eukaryotic cells, and of course, as you all know, this cell is divided into different compartments, and at least all the compartments, well, many compartments that are drawn on this sketch are membrane-bound. So there is a fluid lipid membrane surrounding these organelles, the nucleus here, the endoplasmic reticulum, this blue stuff here, where many proteins are synthesized. The Golgi apparatus, which is this green stack here, where many of those synthesized proteins are modified by enzymes and then sorted. Or here you can also see the collection of endosomes, which form from the results of endocytosis. And these endosomes also are membrane-bound. And what's interesting, in addition to this compartmentalization, is the fact that everything is very dynamical in the cell, obviously. And in particular, you have some transport pathways. So you have these two typical pathways, which is the secretory pathway, which goes from the endoplasmic reticulum to the Golgi apparatus and then to the plasma membrane or other parts of the cell. So this is what the cell gives out, basically. And you have the endocytic pathway, which comes from the plasma membranes through endocytosis and then goes into early endosomes, late endosomes, lysosomes. And of course, this pathway interacts with one another. And so everything is very dynamical. If you were to mark one lipid molecule that is part of one of these membranes, this lipid molecule would basically go away within a few minutes and would be replaced by other ones. And so this sketch here, which seems really static, should be understood that everything is basically interacting with everything here. And this is what is shown in this other sketch, which is a little more detailed. So here you've got the two routes, this secretory pathway and the endocytic pathway. And what this sketch shows, first you see a bunch of arrows, which tells you that everything basically exchanged with everything else, pretty much. What you also see is you have these little colored spherical things here, which are different protein codes, and these are basically the aggregates of proteins that help deform the membrane and creates transport intermediates. So basically, when a coat forms on the endoplasmic reticulum, it's going to make a vesicle, and then this vesicle is going to be transported to fuse with the Golgi apparatus. Similarly, there are vesicles that bud from the Golgi apparatus to fuse with the endoplasmic reticulum, and et cetera, et cetera. So you've got three main kinds of protein codes that do this: the clathrin, which is responsible for endocytosis, and then the COP2 and the COP1, which basically are involved in Golgi transport. And the main question, one important question, in addition to what is the role of the different molecules in allowing these different steps, one important question is how all these arrows directed. Is everything random? Certainly not, because you've got fluxes. So how do basically large-scale fluxes emerge from local interactions between basically membrane components? So all these organelles are bounded by a membrane, so the reason they fuse is probably because there is some sort of molecular recognition between membrane components. So we will basically focus on this. And so this structure should be seen as a steady state of an out of equilibrium system. So the Golgi apparatus is always there, but every molecule moves in and out of the Golgi apparatus, so this is really a steady state with fluxes coming from here and from there. And so this is basically the concept that I want to explore. How do you describe eukaryotic cell organelle dynamics based on self-organized on equilibrium steady states? So looking even a little more into the details of what is an exchange between two organelles. So here we've got a donor organelles, could be the ER, for instance. Here we've got an acceptor, could be the Golgi. And so as I told you, you've got this protein coat that forms, and this coat protein interacts with some cargo. And so when a vesicle is formed, there is the cargo that is trapped inside. And then this vesicle is cut from the donor by some proteins. And so you've got a vesicle, the coat disassembled to form a new vesicle, and this vesicle is then transported often through molecular motors that interacts with the cytoskeleton. When it reaches the acceptor compartment, you've got more local interactions between proteins that leads to the fusion and to basically the transport of the cargo from here to there. So you've got three main steps: the budding of a vesicle, the transport of that vesicle, and the fusion of the vesicle. And all these steps are heavily influenced by what is the composition of the membrane. So the composition could be in lipid, cholesterol, the different proteins, and also in some in classes of GTPases that are supposed to give an identity to the membrane. So one such class is the Rab GTPase family. It's a very large family that have more than 70 members. And if you look at a sort of map of where the different Rabs are located in cells, you realize that different Rabs are located in different organelles. For instance, in the Golgi apparatus, you have Rab6, Rab8, Rab2, Rab1. In the early endosomes, which are there, you've got Rab5, Rab4. In the late endosome, Rab7, etc., etc. So you've got a bunch of different kind of Rabs. And one of the ideas is that these different Rabs, they give an identity to the membrane, and they're going to say when a membrane should fuse with another membrane. So there's a few facts that are interesting about these Rabs. First, they are GTPases, which means that they have two states. One inactive state in which they basically diffuse in the cytoplasm, and then they bind GTP and they become active and they are membrane bound. So when they bind GTP, they become stuck to a membrane, and eventually they hydrolyze this GTP and they become cytosolic again. So you have an activation-inactivation cycle that consumes energy, and each time you consume a GTP molecule, you go basically from binding to unbinding. So this is obviously out of equilibrium. In addition, all these different steps that I talked about, they are influenced by the composition of the membrane in Rab. So depending on the kind of Rab you have, you will select different kind of cargoes, you will be transported in different ways, and you will interact differently with the fusion machinery. So basically, one of the ideas is that two organelles or two membranes that have the same composition in Rabs can fuse with one another, while two organelles that have different composition cannot fuse easily with one another. This is called homotypic fusion, and this is going to be one important component of the model. Another thing that's interesting about the Rabs is that this activation-inactivation cycle is actually sort of cooperative, which means that if you start with a membrane that has a given color, so a blue Rab, let's say, and then some red Rab becomes activated, these tend to inactivate the blue Rab, which will then leave the membrane, and this membrane turns it to red. So you have irreversible maturation of the membrane from blue to red due to the coupling between the activation cycle of one Rab and the inactivation cycle of the previous Rab. So this is called a Rab cascade, and this sort of gives an arrow of time for the membrane identity. So the idea is to try to make models of this really complicated system. Of course, we will simplify this very much. And the idea is to basically have as little ingredients as possible in the model so that we can make sense of it. So it's more of a conceptual model than an actual model, which is supposed to represent all the complexity of the cell. And the ingredients that I want to put is the fact that budding and fusion will depend on the composition of the membrane. That's one thing. And the other is that the composition of the membrane itself will evolve with time. So this is basically budding and fusion are composition specific, and the membrane identity undergoes maturation. And with this, we'll see what we can get. Okay, so some experimental facts. So this is an example of the Golgi apparatus in algae, and you can see that this structure is really amazing. So basically, each of these little bags that you see is one vesicle, which is about a micrometer wide and very, very flat in this case. So it's basically one bag of membrane. And you see that in the Golgi apparatus, these bags are stacked onto one another. And it's a very structured organelle, and nevertheless, as I said, there are fluxes going through this organelle all the time. And the little balls that you can see here, these are the transport vesicles I was talking about. So there's a lot of things going on, but nevertheless, the structure is very well defined. So this is what happens for higher eukaryotes, plants, mammals, and etc. If you look at different kind of organelles, for instance, in yeast, in Saccharomyces cerevisiae, the Golgi is very different. you can see that the different bags here, which are called cisternae, they are dispersed throughout the cytoplasm of the cell. So there's no stacking, and it's not clear that there is an exchange between these bags. Well, here it's believed that the bags themselves, they exchange components. And here, since they are dispersed, you can actually follow one of those cisternae over time. And you can see that one cisternae goes from a color which is a signature of an early identity to a color which is a signature of a late identity. So over time here, the early identity goes down and the late identity goes up. It takes about 1 minute. And so basically the idea is that, so this difference, so early here in the Golgi is called cis, and late is called trans. And if you look at this stack structure, the cis Golgi is basically what is receiving the influx from the ER, and the trans Golgi is where things go out from the Golgi. And the medial Golgi, which is an additional identity here, is what's in between. So you got these three identities. They're supposed to interact by budding and fusion and maturation, and in some case it makes this structure, and in other case it makes that structure. So why? What's the difference? So one difference is that here the time scale is typically 1 minute, while here if you follow a protein going through the Golgi, it takes about 20 minutes. So you've got an order of magnitude difference in time scale between these two species, and you also have a very different structure. So are those things related? The answer, I believe, is yes. Okay, so now looking at the Golgi of mammalian cells, as you can see here, you've got a nice structure, but this is an EM. So here you see the structure, but you don't see the dynamics. And actually, although we've known this organelle for more than 100 years, we still don't know how things move inside the Golgi, and that's one big question. And there are basically two models. One model is the model where those different cisternae are static, and exchange proceeds by vesicles that come from one cisternae and fuse with the next. So in this case, the vesicles are going in an anterograde fashion from the entrance to the exit, and they basically transport cargo from one cisternae to the next. So that's the vesicular transport model. And then you've got the other model where the cisternae themselves are undergoing maturation, they're changing color over time, and this is how proteins basically move and change environment. Here a protein moves because it gets into a vesicle and the vesicle moves. Here the protein moves because the cisternae and the entire cisternae changes color, changes identity, and this is how the protein is going to, or the cargo is going to see different environments. But of course, the different enzymes which are specific of particular color, they have to come back to where this color is. So an enzyme that is carried this way, if it wants to remain in the cis Golgi, it needs to be transported in a retrograde fashion. So in this case, vesicular transport is retrograde from the exit to the entrance, and here it's anterograde from the entrance to the exit. And people are fighting over those two pictures. They've been fighting for a long time, and there's still no clear consensus on what is actually going on. So this is one of the interesting questions. There's another interesting question. So here we're looking at endosomes, and we're looking at early endosomes, so basically the recipient of the endocytosis. And if you follow an early endosome over time, you can look at the size of the endosome, so the typical radius, and you see that it fluctuates quite a bit, but it reaches some sort of a steady state. And then you can look at the color of the endosome. So this endosome is going to be an early endosome, but eventually it will turn into a late endosome. And so if you look at the amount of early endosome marker you have in this endosome, you see something like this. So basically, when there is a fusion with another endosome, the fluorescence goes up, and then the fluorescence goes down, and then it goes up and down, and it fluctuates very much. It's a strongly stochastic variables. And then eventually, the early endosome fluorescence goes down to 0, and this early endosome has turned into a late endosome. And so one question you can ask is, okay, so what's giving rise to all these stochastic fluctuations? And then how much time are you going to have to wait for this thing to become a late endosome? Obviously, it's not a deterministic process. It doesn't decay exponentially. Sometimes it goes up. So it's a sort of first passage time process to know when this endosome is going to turn to a late endosome. And so basically, we've got these two aspects, which is the fact that it's a non-equilibrium steady state and the fact that it's very, very stochastic, and they are probably both important. So this is a sort of model that I want to tell you about. So this model is designed for the Golgi apparatus. So the idea is that you've got an influx, which comes from the ER, and the influx is an influx of blue membrane. So it's a membrane that corresponds to the cis Golgi. And then you have an outflux, which is, let's say, the plasma membrane or wherever things are going. And let's say that this is green, so it corresponds to the late identity. And in between, so you influx vesicles that are blue, and then these vesicles can do two things. They can fuse with one another to create bigger compartments, but they can also undergo maturation from cis to medial and medial to late. And so the question is what is the steady state that you get, depending on the rates at which you import things, the rate at which the things can maturate, and the rate at which they fuse with one another. Okay, so let's first start by describing some sort of basic concept of phase separation. You probably heard already some of this morning. So both equilibrium and non-equilibrium. So this is an example that it has to do with phase separation in membrane, but it's the same if you look at bulk fluid. So basically the idea here you have examples of, this is a giant vesicles. The two different colors are supposed to tell you about two different compositions. And here you can see that you have domains, which means that you have a phase separation into different compartments of different components into different domains. These domains are circular, which means that there is a line tension that wants to reduce the area of the domain. And you can make a phase diagram in terms of your three components in this case, and you find phase separation somewhere here, and that depends on the temperature. And the main difference between it being in a membrane or in a bulk is that in a membrane, if you have phase separation, this can be coupled to the deformation of the membrane, but we won't talk about this now. So how do you describe this? Well, one easy way to describe this is to do a sort of mean field model, where you say that you have a fraction, let's say you've got two components, and phi is the fraction of one component in the membrane, and you write a Hamiltonian or a free energy that includes a bulk energy terms, which has some entropy contribution and some interaction contribution, and it also has some interfacial term that tells you that if you change the composition abruptly, this is going to cost some energy. And so if you minimize this, you realize that there is a phase separation, which means that you go from one phase to two phases whenever the interaction parameter is larger than some value. And that defines basically the spinal decomposition curve. So this is the inverse of the interaction potential. This is the composition. If you're here, you have two phases. If you're here, you have one phase. And so this is a bit complicated, so you can do something simple. If you dip into the two-phase system, you can say, okay, let's forget about this, and let's say that I just have drops. So I have a drop. Inside, I've got one composition, outside another composition. And then my energy is basically going to be simply a difference of chemical potential between being inside and being outside, and some sort of line energy which corresponds to this boundary term here. And so you can describe everything this way, and that's pretty good if you're deep inside the phase-separated regime. Okay, now if you want to look at the dynamics, so here you see the dynamics of coalescence of the different domains that are in a vesicle, and you see that these domains, they diffuse, sometimes they meet, hopefully one is going to meet, okay, so here you see, look, they meet and they coalesce. And so that's one type of kinetics that works by diffusion and coalescence. You've got different kind of kinetics, and basically what you expect is that the radius of these domains should increase with time with some power, which is a dynamical power law here. And depending on how these things grow, you'll have different power laws. So one growth process is called Ostwald Ripening, and is due to the fact that these drops have a line tension. So the smaller drops are basically less favorable than the bigger drops, because bigger drops have a smaller perimeter. So this growth process is going to proceed by the smaller drops shrinking and the bigger drop growing. And you can work out how you expect the radius of drops to increase with time. And so basically you simply say that this thing grows because there's a flux of material. And the flux of material is due to some gradient of chemical potential, which you can sort of calculate by saying that you have a single size in the system. And then the difference of chemical potential is related to the line tension through something that is equivalent to the Laplace law. And if you put that together, you get a growth kinetics, which is like time to the power 1/3, which is one famous growth law. And what you get also is that the size distribution of the drops is going to be peaked for a given size. So you don't have any small drops because they shrink. You don't have any large drop because they haven't had time to grow. And so you have a peak, and this peak moves in time according to this law. So that's one growth process. This is purely equilibrium. There's no energy spent in maintaining the system into any sort of steady state. Another possibility, which we just saw in the movie, is that two domains are going to diffuse, find each other, and coalesce to make a bigger domain. So in this case, the dynamics is a bit different. Basically, you say that the radius increases according to some diffusion process, which depends on the diffusion coefficient. And if you work this out, then you find, if you work this out, then you find that the growth is also a power law, but a different power here, which is 1/2. And what's also very different is the size distribution of the domain is going to be an exponential. It won't be peaked because the small domains, basically, they don't shrink. They are still there. In order for small domains to disappear, they need to find other domains, and this takes more time. So at the end of the day, what you end up is an exponential size distribution. And if you want to do this a little more properly, to actually work out the kinetics more properly, what you follow is you can follow a master equation approach, where you look at the concentration of domain of a given size, and you say that these domains, the concentration evolve in time, because basically these domains are going to fuse with other domains. That's one possibility. And then the other possibility is that these domains will increase in time because smaller domains fuse with one another to form these domains. And so this is basically a kinetics, and if you work out, if you solve this, you find something like that. Okay, so this is equilibrium, and at the end of the day, if you wait long enough, what you should see is two big domains, because this is the lowest energy state. But if you look at real membranes, or as you heard this morning, the cytoplasm of cells, you don't have macrophase separation like this typically. So this is a sort of fake colored distribution of proteins in the plasma membrane of the yeast, and you see that you've got many smaller domains. You don't have one big two-phase region. And so there's many possibilities for this, but one that is fairly attractive is the fact that because this is an out of equilibrium system, you cannot go to the macrophase separation, because permanently these domains will be break into smaller domains for some reason. So one possibility is these domains might be broken by the fact that, at least for membrane things, that they are recycled. So if this is the plasma membrane, you've got an influx due to exocytosis, an outflux due to endocytosis, and what you have in the middle is a steady state that is the balance of these two fluxes. And so to know what the typical size of the domain you're going to get, that will depend on the rate. The faster you recycle things, the smaller domains you will have, because they have less time to grow. And basically, the rule of thumb is that you can take the dynamical scaling at equilibrium, and you replace time by 1 over the off rate, and you find the steady state size of the domain, which now depends on the rates at which things happen. So this is the basic concept for steady state of a non-equilibrium system undergoing phase separation. You don't get macrophase separation, you can have some sort of mesophase separation controlled by kinetics. Okay, it's all clear? You interrupt whenever you want, of course. All right, so, okay, so maybe I switched that, I moved it. So just to tell you that this sort of concept has been applied to various biological systems. So this is the size distribution of cadherin clusters at the junction between two cells in the tissue. And you can see that there is a nice power law with an exponential cutoff. So maybe I should just tell you about that. So basically, when you work out what is the size distribution of domain in this out of equilibrium system with recycling, you use the same master equation. And the steady state in this case is not an exponential. It's basically a power law with an exponential cutoff. And the exponential cutoff basically depends on how fast you recycle things. So the typical size of the domain will be this exponential cutoff, which is given by that, as I wrote before. And then the power law you have here for smaller domain, that depends on how, basically, on what is the size dependence of the fusion and diffusion rate. But if the fusion and diffusion doesn't depend on time, you expect a power law which is minus 3/2 here. So this is a steady state size distribution. And in the case of cadherin, this was sort of confirmed experimentally. And then you also have the size distribution of endosomes. And you also have, so it is a bit more messy, but you also have basically a power law and an exponential decay. So this concept can be applied fairly directly to the size distribution of basically non-equilibrium aggregates in cells. Okay, so let's go back to our problem now, which is finding the steady state of this complicated system. So again, here you have an influx, which is a parameter, the influx. And then what's happening is that you will have fusion inside, and the fusion will be homotypic, which means two things of the same color can fuse, but two things of different color cannot fuse. If you have the budding will also be specific, so when you bud something, you will bud only one color. And then you have maturation. So one cis piece of membrane can turn into a medial, and one medial into a late. And of course, if you have a big compartment and this happens progressively, it means that you can have compartments that have different colors. And these different compartments can fuse with one another depending on how similar the composition is. For instance, here you got one red square, here you got a bunch of red square. So the probability of fusion will depend on how many red square you have here and here. And these are the rules of the games. And this is actually fairly complicated. So first we'll look at two different problems which are important for to understand the complete dynamics of this model. So basically what's happening here is that one question is how many compartments typically will you have and how pure will this compartment be? Will there be single color or will there be a mix of color? And in order to understand whether these compartments are pure, you need to understand what is mixing the different colors and what is separating the different colors. And one way to separate the different colors, this is pretty ugly drawing, but hopefully you can see, is the fact that the different colors, they bud differently. So if you have a mix of A and B, the B's will bud into B vesicle, the A into A vesicles, and then this vesicle will likely fuse with one another, and that's a way you can separate A and B. So this is basically vesicular sorting, which is a way to separate different identity. The way identity can get mixed is because one identity can maturate into the other within one compartment, which is sort of mixing things. But then you can still expect a steady state here, because if you have an influx of an early identity, which is light blue, this early identity is turning into a late identity, which is dark blue, and then the late identity is expelled by vesicular budding, then you can have a steady state. And stochastic fluctuation can basically turn this steady state into a fully mature compartment for which the influx of immature material will be stopped. And so basically, if you look, you can study this system to see whether a compartment will be at steady state or whether it will be basically dominated by the full maturation of the compartment. And so we can quickly look at these two processes and then mix them together into this big, complicated model. So let's first look at vesicular sorting, and here again I'm showing the stochastic fluctuation of the composition of cis identity in an early endosomes. And this is a simulation of this process. So you start with a mix of A and B, and then you remove B's by budding and fusion. But these B's that have been removed by budding, they can fuse back with the mother compartment. And so if you look at these different processes, you basically have the composition of the compartment, which decays over time because of budding, and then increases because everything that budded out fuses again. And so you basically have budding, fusion, and eventually you will reach the situation where those two species have completely separated. And then, since A and B doesn't fuse with one another, these two things will remain at steady state and will not mix again. So this is an absorbing boundary condition of a stochastic process. And so you can ask several things here. You can ask how much time is it going to take to reach this full maturation of this early endosome into a late endosome. And to do this, so you can do simulations, of course, but you can also make analytical models, like simplified analytical model, where you say that the initial composition is going to decrease over time because of budding, and eventually will reach zero. But everything that budded can, with some probability, fuse back with the compartment, which means that you go back to the initial composition. So basically, you have this stochastic process where you decay exponentially and then you fuse stochastically. And eventually, at some point, you'll reach phi equals 0, where this fusion is impossible. And so you can do you can calculate how much time you would need just without any fusion to reach phi equals 0, and that's easy to calculate. That's basically the minimal sorting time. And then you can ask what is the survival probability of this compartment, what is the probability that it doesn't undergo this fusion. And so you can solve all this. And the results, both of the simulations and of the analytics, is that you basically have two different regimes for the time you need to wait to sort this thing. If fusion is very slow, basically the sorting will be due to budding, and it will be a deterministic sorting which happens at a time which is the inverse of the budding rate, this one. This is fast sorting. But now if you turn on the fusion, everything that you've sorted can come back with some rates. And if the fusion rate is large enough, the time for full sorting actually starts increasing exponentially with the ratio of fusion rate to budding rate. So this is a very slow sorting. So you can see here the sorting time as a function of the ratio of fusion to budding rate. Initially, it's independent of the fusion, and when fusion starts becoming important, this sorting time becomes exponential. So you get a fast sorting regime, slow sorting regime with a boundary that you can work out analytically. So that's one thing you have with this sorting regime. So already you see that the competition of fusion and budding can give you a very complicated dynamic, or at least interesting dynamics. And then you can also ask, okay, let's say I've sorted my two systems. What is going to be the size distribution of all these organelles that are now pure A and pure B? And this is, so to solve this, you can write a big master equation where basically these organelles can fuse with one another, so that increase their size, and they can also undergo budding, which decreases their size. And you can calculate this, and you can calculate the size distribution of these things. And this is basically what you get. So these are size distribution. This is the probability of observing an organelle of a given size as a function of the size. And so here these are very small vesicles, basically. And you see again this power law with exponential decay. And in this case, basically, if the fusion rate is very small, all you have are small organelles because they cannot fuse with one another, so they remain small. As you increase the fusion rate, you see the appearance of a macro compartment. So here this is basically a dynamical phase transition again between very small vesicles and one macro compartment, and that happens when the fusion rate is equal to the budding rate. And so basically what's interesting is that the kinetics of sorting is coupled to the structure in the sense that if sorting is fast, you only have vesicles. And if you want a big compartment, sorting will be slow, and which is what I just said. And so you can make some sort of phase diagram where here you have the initial composition, and here you have the ratio of budding over fusion. And if you want to be fast, if you want fast sorting, you will have a very small organelles, and if you want slow sorting, you will have big organelles. And basically, if you want to have as big organelles as possible, but sort them as fast as possible, you have to be close to this boundary here. So there is some sort of optimization procedure for a cell that wishes to have big organelles and sort them fast. And now you can look at some data. So this is fairly theoretical. Can we see this in any experimental data? And here, what I'm showing is data where using some drugs, which is BFA here, the Golgi apparatus was forced to collapse entirely into the endoplasmic reticulum. So at some time, the entire content of the Golgi apparatus collapses into the endoplasmic reticulum. And what they're looking at is how much time you need to wait for the Golgi to form again. And so basically you could say, well, what you need to do is to remove Golgi components from the ER, and this is going to be done by budding. And so the time you need to wait is the inverse budding rate. But actually, the observation is that the time you need to wait, which is about 20 minutes here, is at least an order of magnitude larger than this budding rate, which sort of fits this model if you assume that you're in the slow sorting regime. So basically what we sort of tend to conclude from these experiments is that the Golgi apparatus trying to reform itself after a collapse into the ER is in the slow sorting regime. So Golgi components are budding out, but they fuse back with the ER because the ER has a lot of Golgi identity. And you need to wait for a long time for enough Golgi material to basically come out of the ER without fusing back to be able to form a nucleus that will then grow into a Golgi apparatus. And basically the time you need to wait is an order of magnitude larger than the budding time. And this is sort of consistent with the expected values that we have for the fusion rate, which here is diffusion limited. So two vesicles fuse when they meet by diffusion. And by sort of trying to find estimates for these parameters, we end up with the prediction that from our theory that the fusion to budding rate should be of the order 30, which sort of fits with these parameters. Okay, so this is how do you form something de novo after fusion? Well, you need to sort things, and this could be slow. That's the main message. Okay, so now the second little story is a story about maturation versus vesicular exchange, which is one sort of big theme in the organelle dynamics. So here let's look at a simple situation where you've got an influx of white material, which is early material. And then this white material is going to turn into a grayish material and then into black material. So this is early mixed, this is early identity and late identity. And then the late identity is going to fuse with an exit. And you can imagine two steady state, or two two kinetics for this system. One is that all the material fuses into a big compartment. This big compartment will be mixed because it will have both white and dark color. And so there is an influx of white color in this big compartment, an outflux of black color. This is at steady state, and that's how basically the system sets up itself. So this is a long-lived steady state. The other possibility is that white components, they can fuse with one another to some extent, but at the same time they undergo maturation, so they become grayer and grayer. And when they are too gray, fusion is basically impossible because of homotypic fusion. So this thing becomes isolated from the influx, turns into something fully dark, which then fuse with the exit as a compartment. So here, what fuses with the exit are big compartments. Here it's just vesicles that come out of big compartments. And so if you want to turn it into, if you want to put it into the terms that are used for the Golgi different models, this would be vesicular exchange and this would be compartment maturation. So we also did simulations of this. So basically this is exactly the model. You've got an influx, a maturation, and an outflux. Here the outflux is only of the mature components. And you look at so this is the size of this compartment. This is the amount of A component you have in this compartment. So A components are those ones. And this is the total size. And so you see the total size sort of reach a steady state here, but it fluctuates quite a bit. And the amount of early components into this compartment also fluctuates a lot and eventually turns into 0. This is when this compartment has become completely of the late identity and cannot accept any influx anymore. And so one question you can ask if you want to know what is the dominant mechanism, is it vesicular export or full compartment maturation, you can basically count the total amount of material that came out of this system as vesicles and the total amount of material that came out as compartments, and you make the ratio of the two, and this gives you some sort of output parameter. If the output parameter is 1, it means that the vesicle dominates everything. If it's small, if it's close to 0, it means that the compartments dominate everything. And so to solve this, you can try to do some sort of continuous description where you just assume that these basically you forget about fluctuations. And what you reach, you always get a steady state. So a steady state where the influx balances the maturation flux, which also balances the outflux. And this steady state is characterized by a given size and a given composition. Okay, so this will never give rise to a full compartment maturation. Now you can look at stochastic fluctuation by solving a stochastic model where all the steps are basically occurs independently at given rates. And then you can ask, okay, so what is the time I need to wait to have lost all my A component into this system? And this is something that you can, well, I won't get into the math, but you can basically calculate this time as a function of the parameter, which are here the influx and the maturation rate. And you find something for the output parameter, which is more explicitly shown in a plot. So here you have the maturation rate, here you have the influx, and all this yellow region here is when you're fully dominated by vesicular export, so like this. And all this region here is when you're fully dominated by compartment maturation, like this. And the transition between the two, so the transition between the two is fairly well explained by our simple model. Okay, so the bottom line here is that vesicular export is going to dominate if basically the maturation rate is small, so that anything that undergoes maturation can be removed, and while full compartment maturation is going to dominate if the maturation rate is large compared to the budding rate. Okay, and then you can complexify this model a bit, and what's the most important element of complexity you can add is, so here in this model the influx was constant, which doesn't really make sense because homotypic fusion should tell you that the influx decreases when the red color increases. So if you put that in the model, so basically you say that the influx depends on the composition, then you get a more interesting dynamics. So just to show you what's more interesting, well basically what's more interesting is this. So now if you look at trajectory in the phase space, you can see that this trajectory now start becoming, so there is still a steady state, but you've got trajectories that actually are underdamped oscillations. So basically the composition of the compartment first increase very much in A, but then A starts undergoing maturation. So this decreases A, and this also decreases the influx and increases B. But so and then at some points B is very large, so the export will be large, and so that decreases B, and so the influx will start again, and that leads to some sort of spiraling kinetics here. And during this spiraling kinetics, the compartments spend a long time in a region where there is very little A molecules. So here, stochastic fluctuation can very easily completely remove all the A molecules and you become isolated. So the main difference between having this influx that depends on composition is that first you will have oscillations in the composition. And by the way, if you look at the oscillations, you can sort of see that experimentally. So this is the amount of cis component of early component in an early endosome, and you can see clearly that there are oscillations. So this tells you that there are some nonlinearities in the system, and one nonlinearity is the fact that the influx depends on composition. And what this also does is that it tells you that the isolation time will then become almost deterministic, so this system will be much less sensitive to stochastic fluctuations. All right. Okay, so now let's move into the big model, keeping in mind that we already know two independent processes. One process is the sorting of different components, which proceeds by vesicular budding, basically. And one process is the mixing of components, which occurs by maturation. So now let's mix those two. And we're going to start with simulations. So the simulations will be shown as little sketches like this, bunch of balls of different size and colors. The size of the ball is basically telling you how many vesicles have fused to make one big size like this. All right? Okay. And the color will tell you where you are in the phase diagram. So if you're black, it means you have a mixture of A, B, C, so it's a mixed compartment. If you're blue, it means that it's a purely A compartment, etc. And so these are simulations shown in the case where the budding rate is very small. So if the budding rate is small, you've got big compartments, and they are fairly gray in color, which means that you basically have big compartments that are very, very mixed. As you increase the budding rate, the compartment becomes smaller because budding breaks compartment, but it also becomes much more pure. So there are much more of one color, either blue, green, red, or green. But they're still fairly large. And if you increase the budding rate even more, then you end up with basically a collection of vesicles. This is barely an organelle. This is a bunch of vesicles, which obviously are pure because they are just vesicles. Okay, so now you can put this into phase diagrams. So both the purity, which is basically how far from the center of this three-color triangular plot you are, and the size, which is basically what's the average of this size distribution. And this is a function of the budding rate and the maturation rate. And what you can see is that there is a clear boundary here between pure and impure compartment, and this boundary is basically when the budding rate is equal to the maturation rate, which sort of makes sense because budding sort things and maturation mixes things. And you also have, as you increase the budding rate, the size of the compartment becomes smaller. So if you believe that the Golgi apparatus of mammalian cell is an organelle where compartments are large and fairly pure, which is the way it is usually described, to get this, you need to choose fairly specific values of the parameters where the budding rate is basically close to the maturation rate and also fairly close to the fusion rate, which is what is used to normalize all these rates. If you don't have this, then either you get big mixed compartments or small pure compartments. Since I don't have a lot of time, I won't tell you about the analytics, but we can also understand all this analytically. Now let's look at transport. So remember that there are these two models. Vesicles are either going in an anterograde fashion, if the cisternae are basically fairly static, or if cisternae are themselves evolving, then vesicles should go in a retrograde fashion. And which one is happening is a question. So in order to see which one is happening in our simulations, we basically follow a cargo which is going to move inside vesicles. And we're asking, when this cargo is undergoing a vesicular exchange between two compartments, what is the difference in color of these two compartments? Has the cargo gained or lost the red identity? Has it gained or lost the blue identity? And has it gained or lost the green identity? And if you have anterograde vesicular transport, you expect the cargo to lose the blue identity. The red identity should be roughly the same because here there's a gain and here there's a loss. And so you increase the cargo to gain the green identity. And if the vesicles are going in the other way, you expect the opposite. The green identity should go down and the blue identity should go up. And this is what we get as a function of the budding rate. So at very low budding rates, when the compartments are big and impure, you clearly have anterograde transport, retrograde transport, sorry. You gain blue and you lose green. And when the budding rate is very large, which means that you've got small, pure compartments, then you clearly have anterograde transport where you gain green and lose blue. And what's interesting is that in between, which is basically where I sort of argue the mammalian Golgi should be, what you actually have is a centripetal transport, which nobody talked about before, where you lose both the blue and the green identity and you gain the red identity. So basically what's happening here is that vesicles are not going one way or the other, but they are basically going towards the center of the organelle, which is sort of interesting and unexpected. And the reason for this is, so the reason you have a retrograde transport here is that the maturation rate is relatively fast. So this compartment is fairly mature, and whatever is not matured is going to is what is early identity. So what is matured, when it undergoes budding, it basically fuses back. So there is no net transport. When the net transport is the immature thing that buds and fuses back with the earlier organelle. So this is retrograde transport. In the other limit, it's the other way around. You basically take the mirror image of this. And so what is early is going to undergo some sort of cycle here. What is later is going to bud and then fuse forward. And the centripetal flux comes from the fact that when a red compartment can actually fuse with a blue and a green. And so the red compartment tends to be fairly few in this regime. And so the vesicular transport is dominated by the formation of these red compartments. And actually, if you prevent the compartments to fuse with one another, you see that this centripetal transport that was predicted here disappears, and here you go sharply from a retrograde to an anterograde transport. Right, so so basically, in order to compare this with experiments, it's pretty hard, because in experiments, what usually happens is that you have mutation of proteins that don't work at all anymore, or overexpression of proteins. So what we can only be qualitative, but basically we can see that if you increase the influx, you tend to increase the Golgi size, which is what we predict also, but that's sort of a natural prediction. If you decrease the maturation rate, that's more interesting. You tend to increase the purity of the compartment, which is exactly what we predict. And also, the structure tends to be more stacked, which basically in our mind goes with the purity. So this is a non-trivial prediction, at least. This is a non-trivial result of the model that is also observed experimentally. And then you've got basically a mutation, such as decreasing the fusion rate, which leads to Golgi fragmentation. In our mind, it leads to many smaller aggregates. And basically, okay, so there's several of those. They're all consistent so far, but it all remains qualitative. So to conclude this, the main message here is that only having local interactions between constituents and having some sort of irreversible reaction, which in our case is the maturation of the biochemical maturation, this can lead to steady state fluxes through a system, which is of course the hallmark of non-equilibrium system. And you can make a phase diagram where you relate the structure with the different rates. So this is budding, this is maturation rate, and basically what we predict is that for small budding you have large compartments that are impure, for large budding you have small compartments that are pure, and in between you've got something that's not when you look at it, but basically when you look at the size and purity, this could resemble the mammalian Golgi. And now the sort of provocative proposal is that if you compare two species with completely different organization, although the actual proteins are not that different, then this could basically be explained, these two structures could basically be explained by being at different place in the phase diagram. The mammalian Golgi would be in the sort of well-sorted regime, while the yeast Golgi would be in the mixed regime. And so here you'd have basically anterograde vesicular transport, and here some transport that is much less trivial and could be even towards the inside of the aggregates. Okay, so I'm going to, so what basically we'll be looking at now is to actually put some real cargo in this system and look at the way they are transported, because we can compare this with experiments when they look at the kinetics of cargo transport depending on the kind of cargo. And these are the several things that are not in the model and should be in the model. We should have space. We don't have space so far. Of course, the different compartments can also undergo adhesion, and the exchange between compartments can be related to how far they are from one another. So all this is not there, and this is complicated to put in, but we are thinking about it. And although this model is very simple, I think one thing that it does, it gives boundaries to what a more complicated model could do. So if you want, for instance, to promote anterograde transport, because this is what your system shows, then this is only going to happen if you put some feedback in the system, so that basically you say, for instance, that any vesicle that bud from a compartment undergo maturation after budding, then this is going to promote anterograde transport. If you don't have that, then this vesicle is very likely to fuse back. All right, so I thank you for your attention. Just to tell you that this work was done, most of it, that is the Golgi part, was done by Quentin Vagne when he was a PhD, and by Jean-Patrick Vrel, who's a PhD with me right now. And I thank you for your attention.